So, single cell sequencing data analysis. So, if you are learning bioinformatics or you want to learn bioinformatics, you should know that the single cell sequencing has revolutionized our understanding of cellular heterogeneity and gene expression patterns at a single cell level. So, the Seurat is a widely used R package that provides a comprehensive pipeline for analyzing the single cell sequencing data. So, in this video, we are going to see how single cell sequencing works, what is the basic pipeline of it, and how we can use the R tool to analyze the data of single cell sequencing and where we can get the data of single cell sequencing as well. So, first of all, let's talk about 100x, 10x genomics. 100x genomics or 10x genomics. So 10x genomics is a website that provides the high single cell sequencing data. So in their data sets, if you search for the single cell sequencing, there should be a data set which 10x genomic data sets. So here we can open their data sets and here we can get the data of single cell sequencing. So let's just search for the single cell. So here we can see single cell gene expression, single cell gene expression fracs, and a lot of other values and human data. Also, tumors mouse data is available there. We can check any kind of data that we need for our expression levels. So how this analysis works? First, we have to filter out the important genes. For filtering analysis, we have to figure out these genes and then we have to filter those. After that, we have to normalize the data. And then we have to identify the highly variable features and then scale that data and then get our PC analysis. PC gives us the genes that we need. So it's a quite difficult phenomena and it's an advanced bioinformatics subject of single cell. So if we move on to our Linux terminal here in our folder, you can see if I open my terminal here, you can see I got a HV5 file that we can read. If this is a single cell sequencing data, 40K, 40K. No, we can't read this file. So this file is unreadable. So we have to just quit it and let's just learn about it. And then we are going to use the R code to analyze this. So after getting our PCA done, we can make the clusters of the gene and then we can get the UMAP of that as well. So first of all, install the load and required packages. The required packages is Seurat, ggplot2 and the dipeller plot. And then we are going to import and pre-process our data. So import your single cell data. You can download any type of data you want, but if you're going to use my data, I'm going to give you my name. You can download it from the 10x genomic website. It is a 300 megabyte something file. So it is going to take time to analyze as well. So assume that your data is stored in local data variable. So you have to create your object as well. After creating your object, you have to pre-process and then move on to quality control. So we are going to filter some cells and genes, remove the low quality cells and genes based on the threshold. So we are going to set the threshold above 200 and then we are going to normalize our gene expression data. So normalizing the gene expression data to account for the differences in library size and capture efficiency. And then we are going to identify high variable genes. So identifying the high variable gene varies from cell to cell and focus on informative gene for our downstream analysis. So then we are moving on to the dimension reduction and clustering. So performing the dimension reduction and clustering to identify cell populations and the visualization of data. Uniform manifold approximation and projection UMAP are T-distributed statistic neighbor embedding analysis are used. So we are going to reduce the dimensionality using the UMAP, which is Uniform Manifold Approximation and Projection. And for visualization and clustering, then we are going to perform the PCA. PCA is a highly variable to reduce the dimensionality of data, and then we are going to make the clusters of cells, identifying the cell clusters based on the reduced dimension using an clustering algorithm, which is Lowing algorithm. So we are going to explore the and visualize the clusters of genes. So identifying the cell clusters and then visualizing these clusters and explore the expression pattern of specific gene within the each cluster. Here we got the few options. We can use a dimension reduction plot, and then we can also use a feature plot within the each cluster, and then we can also use the volume plot as well. So identifying the differentially expression gene between the cluster to identify their unique characters, we can find their markers. We can also find their differentially expressed genes. Additional analysis that we can do depending on your research question, you can perform additional research question. For example, trajectory inference, cell type annotation, and gene set enrichment analysis using Seurat functionality. So gene set enrichment analysis, we can also do that to find the pathways and gene set, annotate the cells to find our markers, and also find the trajectories and the development paths. So analyzing the single cell sequencing using the R package and the Seurat package in R is a powerful and comprehensive pipeline for exploring the cellular heterogeneity and gene expression pattern at a single cell level. So applying the Seurat pipeline to your single cell data will help you to gain insight into cellular diversity and uncover important regulatory process underlying the complex biological systems. So that was it for our introduction lecture to the single cell and how we can use the single cell to make the many projections and stuff like that. So in the next video, we are going to use the code to analyze a big data set of 300 megabytes.